Hello everybody, welcome to a video on the innate immunity. So we'll start in on the innate, couple quick videos on the innate, and then we'll move to the adaptive. And the innate immune system, or innate immunity of the lymphatic system, is your first line of defense. So we'll talk about external barriers and then antimicrobial proteins and a little bit about phagocytosis to get us started. So these are your external mechanisms, if you want to call them those, external ways to protect yourself innately. And there's 14 on the side, and we'll match them up to the 14 on the right, no problem there. And then in terms of your sort of molecules or cells, so your molecular and your cellular response of innate immunity, we'll talk about the 10 things on the left, and we'll match them up to the 10 things on the right. So these are now molecules that you use to defend yourself and cells that you use to defend yourself innately. And the human lymphatic system with immune function is very cleanly split between two things: innate immunity and adaptive or learned immunity. We're going to put adaptive immunity to the side for the next few modules coming up shortly and focus right now on innate. And the difference between the two could not be any cleaner. It could not be any clearer. It is specificity. The innate immune system innate immunity does not care whatsoever what it's targeting. It just goes after and it just attacks and destroys. Adaptive or learned immunity is only components that are very specifically choosing one particular microorganism or antigen or virus or bacteria and going after one specific thing. Right? Couldn't be any cleaner in terms of that split between the two. And maybe already you've heard me already start talking about pathogens, invaders, microorganisms, viruses, bacteria. What is all this? It's the good guys versus the bad guys. I go back over these modules and it just staggers me that we're still alive and that we fight so hard against everything trying to kill us on a second by second basis of your entire life. What are the good guys? You talk about cytokines, histamines, and leukotrienes, and things like that. Complement proteins, antibodies, of course, are the difference between life and death, being able to produce the right antibodies quickly. T cells and B cells, the cells that go to work, and natural killer cells, those are the three lymphoid cell line cells that keep you alive on a second by second basis. Macrophages, neutrophils, all that. And on the right hand side, the bad guys, so infectious blank. Anytime you hear me say infectious agents, infectious microorganisms, something that's infecting you that's going to do you harm. Pathogens is another way you'll hear me talk about microorganisms. I'll shuffle these all around. Microorganisms, invaders, bacteria, viruses specifically, fungi, parasites specifically. Antigens is a key word. That's a foreign substance that's going to cause trouble if you don't figure out how to deal with it. And just be looking for this root the whole time. Side with the soft C. C-I-D. And I'm sure if you watch all your crime shows, you know exactly what homicide is. Man, so killing of a man or another human being is a homicide. Insecticides kill insects. Okay, so the siding is the killing part. So just look for that root in there. And you got your good guys trying to kill the bad guys, and the bad guys trying to kill the good guys. And your health and your life depend on who wins this battle. And if you're lucky, for 122 years, these guys will beat up these guys for 122 years. And it's miraculous every second of your life. Every second of your life, it's a miracle. It just anyway. Okay, so two types of innate immunity we can talk about. There's the external, and that's going to be sort of conferred. So this is material that does not penetrate the inside of your body. And that's going to be immunity conferred by two important membranes. You remember there are four membranes in the body. Cutaneous and mucous are the two I have here. You also have synovial membranes, and you have serous membranes. Those are way internal. Those have nothing to do with external immunity. Cutaneous, obviously the skin. You go, wait a minute, what about the mucous membrane? That's lining your GI tract. That's lining your respiratory tract. That's lining your reproductive tract, your urinary tract. Well, inside those tracts, the lumen of the GI tract, that's not you. Okay, I realize it's buried deep down inside of you, but it's not you. Okay, material moves in through the mouth, out through the anus, defecated. That's not you. It has to penetrate the epithelium and get inside your body to be you. Okay, the air you pull down, I realize inside your lungs may feel like it, but inside of your alveoli of your lung is not you. Right? That's external tissue. It's external space. So the mucous membrane lines the external space. You just have to think of the body like it's inside out. And the cutaneous line is the actual external body that we know of. And so cutaneous and mucus, between the cutaneous and the mucous membrane, every surface area of your body is covered with those two membranes. Okay? And that's where you're going to have these barriers that we're about to talk about. Innate immunity from these two barriers, the cutaneous and the mucus. And the cutaneous we know is your skin, and we know that those keratinocytes at the top are just such an unbelievably thick, tough barrier for your skin, right? They're packed tight. They are packed in oil and sebum. We'll talk about that in a second. And it's just a dense, thick, hardy layer that just protects microorganisms from invading you. And they just bounce off this, and this is, of course, the stratum corneum of the epidermis. What happens if a microorganism does happen to take root in here somehow? Well, do you remember the progression? This is the stratum basale, and these cells that I'm putting a line through this layer is mitotically active, and it pushes up, and it pushes up, and these layers flake off. You can see the cell already flaking off. These layers are already flaking off. So even if a microorganism does manage to take root, it won't be long before this set of cells is sloughed off, and the microorganism is sloughed off along with the keratinocytes that are constantly being pushed off. And that's another way just to defend yourself against pathogens. And now the mucous membrane, the epithelium, the basement membrane would be running in dark here. The actual epithelial cells would be here, and then the mucus cells would be all here, blubbing mucus up onto the surface, so this whole surface would be covered, not shown here, but it's all covered in mucus. And that mucus is going to be sticky, and it's going to trap any microorganisms that come in here trying to attack and get inside of your body. This could be inside of the intestinal tract. It can be very much the line of the lumen. I'll put an alpha lumen, the lumen of the intestinal tract, and all the microorganisms in the food you eat, and understand there's billions of them in the food you eat, will be trying to penetrate, but this mucus will kind of grab them. And I love this image. It's not a Tortora image, but I think it's really fantastic. Here are the goblet cells, or the mucus cells, and they're blubbing the mucus out in this layer, and the mucus is trapping the viruses and the bacteria. Okay? And the cilia are then going to beat rhythmically in one direction, and they're going to take this mucus, these chunks and globs of mucus, and the cilia are going to beat, and they're going to move the whole thing away from the body. What does away from the body mean? Well, it could be up to expectorate or spit out if it's in the respiratory tract. It could also be down to swallow, and you're putting all this mucus and these viruses and this bacteria that's in your respiratory tract. You swallow it down, it goes in your stomach and meets up with a pH of 1, and bam, right? These things get destroyed inside your stomach. So that's how you're going to protect there. Other fluids are going to repel invaders. Well, the lacrimal fluid of tears starts laterally and flows across your eye and washes out the microorganisms that are going to land on your cornea and try to colonize your cornea. And also lysozyme, pretty important. We'll talk about lysozyme a little bit as we go along. Lysozyme is an enzyme that has bactericidal properties. It can kill bacteria. So not only are you flowing, it's just the flow of tears across your eyes is going to wash away these microbes. But inside of that flow, inside those tears, you're going to have some lysozyme that's going to actually start killing bacteria too. Lysozyme also found in saliva, perspiration, and nasal mucus, which are fluids that we're going to see here. Sebum is the oil from the sebaceous glands, and the sebaceous glands are going to be outlined as such, and the sebum will be produced out onto the hair follicle, flow up, and just cover the surface of the epidermis, cover the stratum corneum, get inside those layers of keratinocytes on the stratum corneum, and just pack it together, and the sebum will, again, further prevent infection and just an even better barrier, and it's also can retard pathogenic growth. Saliva, again, produced from salivary glands and flowing out across your teeth, across your tongue, across the structures of your oral cavity, washing away pathogens, like not letting them set up shop, washing away pathogens. And saliva also has the lysozyme to kill some bacteria too. And then perspiration, flowing across the skin from the sweat glands, so from the eccrine or apocrine sweat glands. Here is the coiled part of the gland. Here is the duct. The sweat comes up through the pore and washes over the top of the skin, washing away microbes, washing away, constantly flushing. Right? A little acidity, in addition to producing these fluids that are flowing across and washing things away, make them a little acidic. The pH of the skin is kept down around the 4 or 5 range. That could harm a lot of bacteria. Vaginal secretion is slightly acidic. It will retard bacterial growth in addition to the flushing outward. And if you really want to get into crazy acidity, talk about your stomach fluid. If you have any sort of heartburn or reflux, you'll know that is searing, hot, burning, scalding, fatal acid inside of your stomach. And you know that when it comes up and it hits the lining, how badly it burns like pain like nothing else. But you're talking about pH of 1.2, which is essentially near to concentrated hydrochloric acid. And anything that goes in your stomach, all those pathogens you ingest in one bite of food, don't kid yourself, has billions and billions of bacteria in it. It's all good. Enjoy the tastiness in the food and let your stomach acids just destroy all the bacteria and pathogens and microorganisms inside of that food or associated with that food. Okay. And then, so low velocity flow, the lacrimal flow across the eyes, vaginal secretions outward, perspiration and sebum across the integument, saliva through the oral cavity, low flow, but just constant flow, just pushing stuff in the wrong direction, pushing it out of the body or away from the body. Right? What if though? That's nice. Don't get me wrong, that really works. But you got a couple other gears. You got another higher gear. It's because the lacrimal flow, vaginal secretion, perspiration, saliva, sebum. That doesn't require muscular. That's all done glandularly, functionally. And so, what if you want to crank up a little bit of the velocity and put some skeletal muscles and smooth muscles into the equation? Start contracting some muscles, right? Urinary flow, just cleansing the urethra. Okay, high flow of urine will essentially flush out at a higher velocity a lot of microbes that are trying to colonize the urethra. Defecation, same thing. Microbes trying to colonize the rectum or the descending colon or even the transverse or the ascending colon. Okay, defecation, pushing out that material and scouring out and eliminating microbes that way. Tussis, coughing. Again, so you can have the sort of ciliated flow or the ciliated beating of the mucus, but if you really want to speed things up, engage the muscles of the core and the diaphragm and the abdominal muscles and start coughing your way out and creating more velocity. And what if you want to go super high velocity? Well, sneezing, 200 miles an hour, vomiting, diarrhea. Those are high output velocities. And with all three of those, you will be violently expelling infectious microorganisms out of your body, right? Out of your body, which is why transmission of infectious organisms through sneezing, obviously respiratory, obviously COVID, and then norovirus, intestinal viruses through vomiting and diarrhea are very, very, very common too, right? So, okay, that's all external, and you got everything protected, but, you know, not everything works out the way you want it to work out, and through the intestinal tract, through the respiratory tract, things can penetrate, or things go real bad, you take your integument and you just put a huge break in your integument. Cut a bagel in your hand, all right? Get a little burn, something, cut, nick, whatever. Scrape a braid and tear away your integument or your mucous membrane, and now you've got an easy path for billions upon billions of microbes to come in and invade. So now what are you going to do? Now you're going to play defense a totally different way. Still innate and still non-specific. You don't care who's coming in and invading. You're just shooting first and asking questions later, right? What are you going to do? I got 4 things that you can do down here. We'll talk about 1 and 2 in this video, move to 3 and 4 in the next. They're all part of innate. They're all non-specific. They all couldn't care less who's coming at them. They're just defending like crazy and just shooting like crazy and attacking like crazy, right? 1, throw out some non-specific antimicrobial substances at the invading armies. Just start throwing chemicals out there going to kill, and they don't care who they kill or what they kill. 2, send out your neutrophils and your phagocytes and get them to start going and fire up your natural killer cells. Just start eating microbes like crazy, non-specifically. All right, number 1, what are some molecules you can throw at these invading armies? Interferons, so important. You'll see them abbreviated IF or INF, so important. They're signaling molecules. They work in a paracrine manner. So this is cell A on the left is going to signal. You can see the interferons. I'll just put an arrow on each one of them. It's moving across to the second cell B over here. Remember, anytime cell A sends out a signaling molecule acting in a hormonal way onto a neighboring cell, that's paracrine. Right? So what's going on here? Any cell that's infected with a virus is in trouble. It's in trouble. The cell's going to die. There's nothing you can do. That cell's done. But before it goes, It has the wherewithal to crank up interferon genes and produce a bunch of interferons, and on its way out, it releases those interferons over to cell B and any cell in the neighborhood. Those interferons bind to a receptor, they're pulled in, and that allows the second cell over here to create a bunch of genes. Those genes are not going to prevent infection of the second cell by the virus. The virus is going to infect the second cell too, but it's not going to kill the second cell. Okay? It's going to come in, and there'll be genes that are available and proteins that are available through the interferon signaling. There are going to be new proteins available through protein synthesis that are going to allow you to kill the virus. So the virus will infect the second cell, but it won't be able to replicate and destroy the second cell. So the first cell is taking one from the team, using interferons to signal all the neighboring cells to protect themselves. Once that first cell, this lymphocyte or this macrophage or this fibroblast that's been infected by a virus, will be able to protect its neighbors. That's pretty cool. That's taking one for the team right there. Complement. Complement proteins, sometimes just abbreviated as complement. A bunch of proteins, and they just do 3 different things that couldn't be any more different, but they're all about killing microbes. They literally couldn't be any more different. 3 roles to complement. You should be familiar with them. One is an incredibly important term called opsonization. Here's your microbe, your bacteria, whatever it is. It's got this little molecule on the side, this red piece sticking out. It's a surface protein like any other organism in the world. The C3, the complements are called C1, C2, C3a, C3b, C4, C5a, C5b, C6, C7, C8, C9. I think there's a C25, a C28. Don't stress the details of the numbers. Just know that this one right here, you don't even have to know it's C3b. Just know that a complement protein can bind to that surface protein, and what that's doing is it's opsonizing. It's targeting that microbe and making it infinitely easier for a macrophage or a phagocyte right here to come and eat the microbe. And here you can see the microbe tucked inside of the phagocyte and secured away and eaten and killed. And we'll talk about that process, how that all works next module too. But don't underestimate the importance, and we'll see all the time, opsonization, right? If you want to think about any sort of way to approach this, try to think about getting a roughly healthy food, like even in the old days when we used to, kids with snack and teaching elementary school, getting kids snack, rice crackers were a super healthy deal. You couldn't really get a young child to eat a rice cracker if your life depended on it, unless you slathered something on the top, jelly or peanut butter or honey or something like that. That's opsonization. That's putting something on top of something to make it tastier and easier to eat. That's what this complement is doing. The complement is going right on top of this microbe and making it easier to find and easier to eat for your phagocyte or your macrophage right here, which is this big green cell. It's just making it easier, and you're just really increasing the chances of this microbe being eaten and killed by your phagocytes. So. Step 2, chemotaxis, called positive chemotaxis. Again, the C3b binds, and when the C3b binds, the C3 binds, this C3a piece comes off, and it kind of floats in the area. So C3 binds, I'm going to show you, they're trying to show you the C3a was the second part right here. No big deal. When the C3 binds, the C3b stays, the C3 part pops off, and same thing with C5a. What does that mean? Well, you have all this C3a and C5a floating around this microbe. That's positive chemotaxis. Here are your neutrophils right here. They're coming in. Let's say there's a blood vessel up here. Here's your microbe over here. These neutrophils squeeze out of the blood. They're ready to go. They show up. They're warriors. What are they going to do? Well, you know what they're going to do. They're going to look around and try to figure out what in the world they're battling. What does C3a and C5a do? They just whack you in the shoulder. I get whacked in the shoulder. What do? I turn and look and see. That's what C3a and C5a are doing. They're binding onto these neutrophils from the direction that the microbe is coming in, and this neutrophil right here just went what? And turned and looked completely to the microbe, because that's the side that the C3a and the C5a bound to. If the C5a and the C3a bound to this side, the neutrophil would head that way. Chemotaxis. Positive chemotaxis. Positive meaning in the direction. Chemo, chemical, taxis, touch. Positively touching towards the chemical, towards the direction you want to go. Pretty cool. And if that's not cool enough, what complement has in mind, a third thing, again, C3 binds in this whole composite collection of other C3, C5, C6, C7, C8, 9. They break off this one piece, they form a pore, and they embed themselves in the membrane. And I'll a much better picture down here. Okay? Much better picture. So this is a complex of complement proteins. It pops off of the microbe, and then it turns and goes inside the microbe, makes a pore that penetrates the membrane of the microbe, and that allows water and fluid and ions to flow in and out, and you end up lysing the bacteria or the microbe. You end up bursting it. Okay? You poke holes in it. This is called a MAC, a membrane attack complex. So it's these chunky complement proteins that get together to form a pore. They stick right in the membrane of the bacteria or the microorganism, and that basically is like poking a hole in a balloon. All the air will flow out, and the balloon's dead. The same thing. They poke a hole in the microorganism, all the fluid flows out, all the ions flow out, and the organism eventually dies, and that's called a MAC. So you're lysing the microbe, or you're bursting it through this process. So anyway, 3 cool ways that complement works. A third thing you can throw, besides interferons, besides complement proteins, throw some iron binding proteins at them. But here's the crazy thing. The iron binding proteins are just doing their job in your body. What are they doing? Well, remember this from erythropoiesis and breaking down used blood cells. So what you have here is all these proteins starting with hemoglobin inside of the red blood cell, moving to transferrin, moving to ferritin. What are they all doing? They're all tying up iron. Hemoglobin is grabbing every iron so you don't become anemic. Hemoglobin is grabbing every iron atom in your body it can find. Transferrin is grabbing every iron atom in your body it can find. Ferritin just grabbing every iron atom it can find and binding them up for use in your erythropoietic pathways, in your red blood cell pathways, and your hemoglobin pathways. And what's that doing? It's limiting iron from these microorganisms. You say, okay, I got infected at the site of infection by 1 million bacteria in this tiny, tiny little pinprick cut. 1 million bacteria invaded me. Okay, I go septic and I'm in a coma because I went septic. That invaded my whole body. Well, that means that 1 million turned into 100 quintillion bacteria. That's a lot of iron those bacteria are going to need to grow each time and to build and duplicate and replicate themselves mitotically. So if you can hold off on the iron, which is functionally, why iron? Because functionally iron is functionally the most limiting molecule in the body, or the most limiting element in the body. Right? And so if you're anemic, you know what I'm talking about. If someone's anemic, they'll know exactly what I'm talking about, because the iron is so limiting to them that they can't perform physiological function. Now you're trying to turn the tables and make the iron so limiting to the bacteria or the microorganisms that they can't function. So that's what iron binding proteins do. And lastly, throw some AMPs, antimicrobial proteins on them. Super broad spectrum, a bunch of things you can throw at them. They kill in different ways. I love this image because it shows the bacteria on the left treated with antimicrobials. See them just cringing up and dying on the right. Don't worry about the mechanisms. Don't worry about the names. Worry about the root right here. Oh, dermicidin, cathelicidin, thrombocidin. Just pick out that root and know what's going on here. Very broad. You're not responsible for what the AMPs are, what the details are, how to name them. Just know that they generally kill microorganisms. Okay, those are all the things you can throw at them, all the molecules you can throw at this invading army. You can also get to work with your phagocytes, cells that eat. Phago is a root for eat. Phago means eat. Cyte is a root for cell. Couldn't be more simple. Cells that eat microbes. Okay, neutrophils, macrophages, and natural killer cells. We'll start, and here you see these microbes coming in, and we'll start with the natural killer cells. So where do the natural killer cells come from? They are the third type of lymphoid cell. So off this pluripotent stem cell, you're going to have the lymphoid stem cell line. We've briefly talked about your T's and your B's, and that's adaptive immunity right there. We're going to hit that so hard with adaptive immunity. But over here, 5 to 10% of your lymphocytes off this lymphoid cell line are going to be your natural killer cells. Okay? And they're non-specific. They don't care. Okay? They bind to pathogens and they bind to microorganisms, and they don't care who it is. They just bind to anything that has an abnormal plasma membrane. Anything that has a plasma membrane that has sort of foreign or unrecognized proteins, these natural killer cells will bind to them. And when the natural killer cell binds to it, they'll release granules into the cell, into the foreign invading cell, or into the target cell. The granules will either contain perforin, which is a protein that's going to create perforations, just like a MAC, just like I showed you with the membrane attack complex. But in this case, the perforins just are proteins they embed in the microorganism or in the pathogen cell membrane, and they just start poking holes, and they cause cytolysis, cytolysis. You put the emphasis on the T, cytolysis, cell bursting. Okay, and that's what perforins do. Or those granules, they introduce chemicals that create that basically. They induce apoptosis, right? So granzymes, the granules will inject granzymes into the target cell, and the granzymes will go in and they will trigger the apoptosis, or they'll trigger that target cell, that microorganism, to kill itself. So really potent ways, okay? And then your phagocytes, I'll go into this a little bit more detail next module too, a little bit, but your neutrophils are first and fastest to get to the site of infection. Remember diapedesis, there's your neutrophil right there with the multi-lobed nucleus. And diapetesis, really quick, we did this in 40B, just a quick review. When a tissue is damaged tissue here, it releases selectins. And the selectins are these black springs, and they bind to the endothelial cells. There are integrins on the neutrophil. The integrins contact as they're moving along. They contact and they bind to the selectins, and that slows down the neutrophil. And it marginalizes the neutrophil. It puts it on the edge of the capillary. And it slows it down so that it can eventually squeeze through the intracellular clefts of the endothelial cells. And that's going to allow for diapedesis. That's going to allow the neutrophil to get into the tissue and start attacking inside the tissue. Right? And those are wandering macrophages. So those are neutrophils. The macrophages are monocytes that do the same thing. They come in as monocytes and then they develop into macrophages. So either neutrophils or monocytes. Those are the two. Neutrophils come in and they act as neutrophils. They're the first. They're the fastest to attack. Your monocytes come in. They develop into macrophages, which are really big. And your macrophages just clean up everything in sight, right? And those are wandering macrophages. You also have fixed macrophages. Don't forget your fixed macrophages. Histiocytes, which Tortora is now at 22 chapters in, is calling these macrophages that sit inside a loose areolar connective tissue. Inside a loose areolar connective, you have macrophages that just sit, first line of defense. Those are called histiocytes. Microglia, inside of the parenchyma of the brain and nervous tissue. Microglia are phagocytotic cells that are sitting here eating up any invaders that get into the parenchyma of the brain. How important can that be? And don't forget alveolar. Remember, any pathogen that gets inside the alveoli, any dust particle or foreign particle that gets inside the alveoli is going to run head to head with this alveolar macrophage. It's going to eat it and pull it into the cell and get it out of the alveolus. Right? That finishes up. Let's just crank this out. These are going to be the external ways, and then we'll move to the molecules and the cells. So, okay, peristaltic movement of the colon and the rectum to expel feces. That is defecation. 11. Violent, rapid expulsion of feces from the GI tract, and that's going to be diarrhea, which is going to be far more powerful for removing microbes, the lower GI tract. Sebaceous glands in the dermis, it coats the cutaneous membrane. That's sebum. 6. D, violent, voluntary, involuntary contraction of the muscles to propel substance out of the respiratory tract. That's tussis, or coughing. E, moist, mucus-covered epithelium in the GI tract. Okay, so that's going to be the mucous membrane. So that's a big one, 2. E is 2, right? The mucous membrane lining the GI, the respiratory, the urinary, the reproductive tract, and allowing you to clean those areas with the mucus and with the cilium, sometimes cilia, but always mucus. F, also containing lysozyme from the sudoriferous glands. That's going to be perspiration or sweat. G, violent explosive contraction, propel material out of the upper GI tract. Oops, vomiting. And that's going to expel a lot of microbes. H, slightly acidic flow of fluid from the vaginal canal. That's going to be vaginal secretions. And again, not only the flow, but also the acidity, slightly acidic, which will retard or inhibit a little bit of microbial growth. I, explosive involuntary muscle contraction to rapidly expel fluid from the respiratory tract. Okay. Sneezing. Okay. Sneezing. That's pretty rapid. J, production and flow of tears across surface eyes. That's lacrimal flow. K, flushes microbes from the urethra, retards microbial colonization. That's going to be urination or urine flow. L, keratinized, stratified. That's going to be your cutaneous membrane. M, incredibly acidic fluid. That's gastric juice, right? Inside your stomach. pH of 1.2. It'll burn your skin. You put gastric juice on your skin and count to about 10, you'll feel a burn. You put it on your clothes, wait about 3 minutes, you'll have a hole in your clothing if you have gastric juice on your clothing. A flow of lysozyme containing fluid from the salivary glands. That's obviously saliva. Saliva flow. Okay. Done there, and we'll go to the next one. Now it's going to be the molecules and the cells. Interferons are going to put myself here. Or I'll do it the other way. A, from the blood plasma. Proteins combat microbes by opsonizing microbes. That's complement proteins. 2. Okay, complement and inducing positive chemotaxis and lysing through MAC or membrane attack complexes. B, 3 lymphocytes designed from the lymphoid cell line, and they attack any cell. These are natural killers. So B is going to be natural killer cells. 5. C, one of the fixed macrophages embedded in the extremely abundant loose areola. Those are called histiocytes. Basically macrophages that are constantly inside of loose areola connective tissue. D, random assortment of proteins with a broad spectrum of antimicrobial. Those are antimicrobial proteins. Pay attention to the side part of dermacidin, cathelicidin, thrombocidin. E, derived from monocytes. That's where macrophages or wandering macrophages come from monocytes. Huge phagocytes free to travel to the site of infection and just lay waste to pathogens by eating them. F, one of the fixed macrophages within the neural tissue of the brain. Those are microglial cells. G, secreted by lymphocytes, macrophages and fibroblasts. Infected by viruses. Those are interferons. How to protect your neighbors when you're being infected by neighbor cells via paracrine mechanisms. H, one of the fixed macrophages found inside the alveoli. That's alveolar macrophages. The name tells it all. I, most plentiful phagocyte. Those are neutrophils, first and fastest there. 6. And J, just doing their job, are iron-binding proteins, transferrin, ferritin, hemoglobin. But in their odd way, they retard microbial growth by limiting iron. So again, sorry these are running a little long. We'll move into finish up inflammation and fever and on the innate system, and then we'll move into the adaptive system. So again, thanks so much for sticking with it, and I really appreciate it.